Hello students, today we are going to discuss in detail on how the introns which are present in the case of mRNA is spliced. Normally, in the case of the eukaryotic system, the message which is found in the case of which codes for amino acids are not continuous in nature, the gene which is present in our system is split gene. That is, what is split gene means? The gene is consisting of intron as well as exons. What is intron? The intervening sequences are called introns and which doesn't code for any amino acid, whereas exons are the gene that codes for the amino acid. Most of the eukaryotic gene containing segments of coding sequences, exons, that are interrupted by the non-coding sequences, intron. Both intron as well as exons are transcribed by the RNA polymerase II as a primary transcript. We call it as h-RNA, that is heteronuclear RNA. So, in your examination, they may ask you primary transcript or h-RNA. The split gene in eukaryote was discovered in the year 1977 by Philip Sharp and Richard Roberts. During the year 1993, one day they were awarded Nobel Prize for this split gene and RNA splicing mechanism. Here is the diagram which shows the formation of the split gene and the RNA DNA hybrid, how the intron is present, how the introns are removed by the splicing mechanism. Splicing means cutting and removing. Here the table shows the mechanism of RNA splicing. In the eukaryotic system, we have four different types of introns are available. Group 1, Group 2, Group 3 and Group 4 introns are available. And Group 1, 2 and spliceosomes are associated with the mechanism. Group 1 introns are found in the case of the nucleus, mitochondria, chloroplast gene, which encodes for the m-RNA, tRNA as well as rRNA. Group 2 introns are found in the case of the mitochondrial and chloroplast gene, fungi, algae and plants. Spliceosomes are found in the case of the nuclear genes of the eukaryotic system, and it is capable of alternate splicing to create multiple products to give the transcript. So group 1 introns are as self-splicing. Self means it itself will do the splicing mechanism. It doesn't require any additional factors additional factors needed for the splicing mechanism. Here in group 1 intron, a free guanosine residue, don't forget, a free guanosine residue is necessary for the splicing mechanism that is involved in removing. Just it acts like a scissor in cutting the 5' and 3' end of the intron, and it is throwing off and the exons are joined together. And group 2 introns are spliced by a special mechanism which is involving a nucleotide, a special adenosine residue which is present in the conserved region or within the intron itself. That is internal part, that is middle region of your intron contains a special sequence in which a special adenosine, it is having a speciality which is acting as a nucleophile that is responsible for carrying out a nucleophilic attack on the 5' as well as in the 3' end of the 5' end of the intron. So this is the mechanism which is followed. Whereas in another set, in the higher eukaryotic system, it make use of the snRNA. The snRNA is called as Taylor RNA. What is the job of Taylor? The Taylor marks the unwanted portion and it cuts the portion and it stitches. Same manner, the introns are considered as unwanted portion that are cut out and the exons are joined together by the phosphodiester linkage, that is done by the help of the snRNA, small nuclear RNA. And associated with the nucleoproteins, we call it as SNPs. These SNPs are responsible for the splicing mechanism in the higher eukaryotic system. They are also called as catalytic snRNA. Let us see the group 1 splicing mechanism. Here is the diagram which shows the group 1 splicing requires a free guanosine nucleotide. The free guanosine nucleotide is acting as a cofactor, and how it derives the energy, it's not yet clear. The 3'-hydroxyl group of the guanosine residue is acting as a nucleophile that first carries out a nucleophilic attack at the 5' end of the intron. So the 5' end of the intron is cut so that the 5' end of the intron is associated with the free guanosine residue. Now the exon 1 is created with a free hydroxyl group. A new hydroxyl group is created at that region. Then by breaking down of the phosphodiester linkage, the new hydroxyl group now carries out a nucleophilic attack on the 3' end of the intron, so that the exons are spliced together and the intron is eliminated, in which the 5' end is associated with the guanosine residue. Here is the diagram which shows the group 2 splicing reaction. In the group 2 splicing reaction, the 2'-hydroxyl group of the adenosine residue within the intron containing conserved sequence is acting as a nucleophile. The hydroxyl group of this special adenosine residue attacks the 5' end of the intron containing, that is the end of the intron, 5' end of the intron contains a guanosine residue. It is carrying out an attack on it so that the 5' end is attached with the adenosine residue. We can see from the diagram, and the exon 1 is created with a free hydroxyl group that carries out a nucleophilic attack on the 3' end of the intron, so that the intron is eliminated in the form of a lariat, that is a circle with a tail. At the same time, the exon 1 and exon 2 are joined together. This mechanism is continued in the group 1 as well as group 2 introns are continued until all the introns are eliminated and all the exons are joined together by this mechanism. Whereas in the case of the higher eukaryotes, as it is associated with the spliceosome, that is a small nuclear RNA and proteins are associated, we call it as SNPs, or spliceosomes are responsible for the splicing mechanism. In eukaryotes, it follows certain rules, that is most of the introns in the eukaryotic system, higher eukaryotic system, they contains GUAG. What is GUAG rule? It follows GUAG rule, means the 5' end of the intron contains GU and 3' end of the intron contains AG. So how it identifies, how it differentiates whether it's an intron or exon, means in the case of the higher eukaryotic system, all these are nucleotides only. So how to differentiate means always the 5' end of the intron contains GU and 3' is having a AG. This is how it identifies. Then once the GUAG is identified, the smaller nucleotide RNA, that is SNP particle spliceosome, comes and joins to the position of the 5' end and follows the splicing mechanism. The SNP particles, dozens of non-SNP particles are also associated in the splicing mechanism. Together we call it as spliceosome. So how many SNPs are present? In the case of this, means each SNP is made up of 100 to 200 nucleotide length in nature. The snRNA, there are 5 different snRNA are involved in the eukaryotic system. Especially they are found abundant in the case of the eukaryotic nuclei for the splicing mechanism. What are all they means? U1, U2, U4, U5 and U6. Don't forget U1, U2, U4, U5 and U6. Here is the diagram that shows the splicing mechanism mediated by the spliceosome. So the pre-mRNA or hnRNA, we can see the intron is having a splice site. The splice site of the intron is having 5' end 3 GU and 3' end is AG. So whatever the AG, first occurrence of AG is considered as the 3' splice site. So initially the BBP, that is base binding protein, that comes and joins to the special adenosine which is present in the intron, that is followed by another protein called as U2AF35. That is BPB binds to the branch point, we call it as the special adenosine residue region is called as branch point, that is attached by SF1, and the polypyrimidine track by the 2AF65 comes and joins to this position, and the AG is dinucleotide is joined by U2AF35. Once the complex is formed, the U2 comes and joins and engages. So U1, initially U1 comes and joins to the 5' splice site, then followed by the branch point by identification by SF1 that comes and joins to the special adenosine residue, that is BBP SF1, and the polypyrimidine tract is attached by U2AF65, and then AG dinucleotide is joined by U2AF35. Once the complex is formed, the U2 comes and joins and engages the branch point by a RNA base pairing mechanism. So this comes and joins in association with the SF1. Once this is formed, then the catalytic complex comes and joins to this position. This complex is made up of U4, U5 and U6. The a small nuclear ribosomal particle forms the complex, B complex we call it as. Don't forget what is B complex. The U4, U5, U6. Once the B complex is arranged in this position, the catalytic activity is carried out. During this rearrangement of interaction, U1 and U4 snRNA are lost and U6 snRNA is brought into contact with the 5' splice site. The 5' splice site is cut by the U6 snRNA. Then further the splicing mechanism, the newly created 3' hydroxyl group carries out a nucleophilic attack on the 3' end containing AG. So this AG is removed and the intron is removed in the form of a circle with tail, we call it as lariat structure. Both the exon 1 and exon 2 are joined by this mechanism. This is how the spliceosome happens. So what is the rule followed? GU-AG rule. The GU is 5' site of the intron and AG is 3' splice site of the intron. A special adenosine occurs at the middle of the intron is called as branch point, that is attached by SF1, that is also known as BPP, branch point binding protein. Then polypyrimidine tract is attached by our U2AF65, and the AG, that is 3' end of the intron, is attached by U2AF35. When this complex is engaged, the U1 snRNA is at the 5' splice site. Then once this base pairing has happened by RNA binding, then comes the trimeric complex, that is U4, U5 and U6, that is called as B complex. This B complex comes and joins to the branch point region. Then slowly the U1 and U4 is eliminated by this mechanism, then which by activating U6 snRNA. This U6 snRNA is creating a cut at the 5' splice site. Then once the 5' splice site is cut, the exon 1 is created with a free hydroxyl group. That free hydroxyl group carries out a nucleophilic attack on the 3' containing AG region. So the intron is removed in the form of lariat. We call it as a circle with tail, and exon 1 and 2 are joined together by this mechanism. Once the mRNA containing all the introns are removed in the eukaryotic system, it is added with cap and tail, because your RNA is a single stranded one and it is easily attacked by the nucleases, because the reason is that it considers our system is always protective in nature. The invading bacteria and viruses, they inject only the single stranded RNA into our system. So automatically what happens, it is always alert in nature, it tries to attack the single stranded RNA. So how it differentiates whether it's our own RNA from the prokaryotic or infectious organism containing RNA means by the presence of cap and tail. So immediate after removal or the splicing mechanism of introns, the mRNA of the eukaryotic system is added with 5' with a cap, that is 7-methyl guanosine cap, and 3' is attached with the poly A tail. So the capping process we will see now. The synthesis of cap is shown at the 5' end of the pre-mRNA. Normally the 5' end of the cap of RNA is called as 7-methyl guanosine cap, or in short we call it as M7G cap. So how means it is modifying the 5' end of the eukaryotic mRNA containing nucleotide. That is, initially a triphosphate is found in the case of the 5' end. That triphosphate is removed by the phosphohydrolase. That after the removal of the phosphate group, it is a guanosine residue is added, that is a GTP is added by guanyl transferase. During this mechanism a pyrophosphate is removed. Once the newly added guanosine residue is added by the triphosphate linkage, then comes the role of guanosine 7-methyl transferase. What is 7-methyl transferase means? The 7th position of the guanosine residue is methylated. Here comes the role. Here we can see in the next diagram the 7-methyl guanosine is added. The 7th position added with the methyl group, and this guanosine residue is attached to the 5' end by the 5' triphosphate linkage. It's an unusual. Normally the phosphodiester linkage is made up of 3' to 5' phosphodiester linkage, and moreover it is only 2 phosphate. Whereas in the 7-methyl guanosine cap, the first nucleotide 7-methyl guanosine is attached by a 5' phosphodiester linkage. Once this is formed, how this addition of methionine methyl group is taking place means by the 2-O-methyl transferase. So the donor of the methyl group is nothing but here S-adenosyl methionine. This S-adenosyl methionine, in short we call it as Uncle Sam, that donates the methyl group. After the donation of the methyl group, the homocysteine amino acid, the methionine amino acid gets converted to homocysteine amino acid. So further what it is doing, the 2-O-methyl transferase, it also methylates the nucleotide that is next to 7-methyl guanosine, that is first base as well as second base are also methylated. That is 3 such nucleotides are methylated by this mechanism. It's a critical role and it is important for the recognition of the eukaryotic ribosomes to identify our mRNA, and as well as it protects the mRNA from the RNases. Next comes the protection of the 3' end. The 3' end of the RNA is added with a poly A tail by the covalent linkage mechanism. In the eukaryotic system, with the exception of histone, all the m-RNA molecules are added with poly A tail at the 3' end. Why the difference? It's not clear. With the exception of histone, histone is not added with a poly A tail, whereas all the other mRNA of the eukaryotic system is added with a poly A tail. Poly means many adenosine residues are added. Why this poly A tail is added means it is helping in protecting the mRNA by the degradation by the exonucleases. The polyadenation is a very important step for the transcription termination, and it is responsible for the export of mRNA from the nucleus to the cytoplasm for further translation. Normally, the polyadenation occurs during and immediately after the transcription of the DNA to RNA through the action of exonuclease complex associated with the RNA polymerase. After the mRNA has been cleaved, around 250 adenosine residues are added to the 3' end of the cleavage site. This reaction is catalyzed by the enzyme called as polyadenylate polymerase. So, how many residues are added? By the polyadenylate polymerase means around 250 adenosine residues are added by this polyadenylate polymerase. Here we can see that from the DNA you can see exon 1 and 2 enter introns. By splicing, the introns A and B are eliminated, and capping is done for the pre-mRNA and polyadenylation. Excision of the introns causes splicing mechanism, and we can see that the mature mRNA contains 5' end contains 7-methyl-guanosine cap and 3' end with the poly A. Then only it is transported to the cytoplasm for translation by the ribosome. This micrograph which shows the RNA polymerase II is engaged in the development of primary transcript, or we call it as the nascent transcripts, then spliced transcripts and loop of the introns, the lariat formation, all these electron micrograph showing the details how actively the spliceosomes are engaged in the splicing mechanism to form the lariat introns for the process of completion of the transcription. Next we will discuss about the role of RNA, that is rRNA synthesis by the RNA polymerase I. Already I told you we have 3 different types of RNA polymerase: RNA polymerase II, RNA polymerase I as well as RNA polymerase III. Already we discussed in detail about RNA polymerase II, which is responsible for the production of the mature mRNA. Now we are going to discuss about how rRNA is synthesized by the RNA polymerase. The RNA polymerase I is responsible for the production of all rRNA genes except 5S rRNA. Where it takes place, the rRNA synthesis occurs only in the nucleolus. Don't forget, mRNA in nucleus, rRNA always happens only in the nucleolus. It is rich with their nucleolus. In the eukaryotic system, the most actively and productive genes which are transcribed most of the time are nothing but our ribosomal RNA genes, because the 90% of the total RNA found in the eukaryotic cell is nothing but our rRNA, that is found in the region of the nucleolus. The rRNA genes are mainly organized into a single transcriptional unit. Single means we have 18S, 5.8 and 28S. All these rRNA are not individually synthesized. It is synthesized continuously and it is interrupted by the spacer nucleotide. Here we call it as the nucleotides which separates one rRNA from the other RNA is called as spacer nucleotides. It's a continuous transcript, we call it as. The precursor is further processed to individual rRNA, that is 18S, 5.8 and 28S. The transcription of the rRNA takes place in the specialized structure of the nucleus called as nucleolus, where the RNAs are transcribed and combined with the protein to form the complete ribosome. Here is the promoter structure of the RNA polymerase I for the synthesis of rRNA. So, a core promoter region exists between your -10 and -100 region, and an upstream control element is found in the case of the -100 region. We call it as UCE, and core element is found in the that overlaps at the -10 to the start point of our transcription. The various factors, upstream element binding factors comes and join to the UCE position. So what are all the things that happens? In the case of the transcription, we will see in detail. The initiation of rRNA transcription involves the recognition of the upstream element region UCE, then core region attracts the selective factors SL1, that is 3TAF, that is Tata box associated factors. What are all the Tata box associated factors means? TPB, that is Tata box binding factor, that positioned at our Tata box binding protein, as well as SL1. This assembly now brings the RNA polymerase to come and join to the upstream control element region. Further, UBF1 that binds only to the core region but also to the UCE region that comes and joins. The binding up of UBF1 it creates a protein-protein interaction, thereby the DNA loops and it activates RNA polymerase I complex for the start of transcription. The RNA polymerase I escapes and clears the promoter, and UBF1 and SL1 further remains in the promoter region itself and ready to recruit another polymerase I. In this way, each active rRNA or genes are transcribed multiple times simultaneously. So, an additional factor TIF1C also stimulates overall rate of transcription and suppress passing of the polymerase I. So, in all the genes of rRNA, the polymerase, 3 individual RNA polymerase comes and joins for simultaneously by this looping mechanism, and they are get transcribed. This is promoted by, the elongation step is promoted by the factor called as elongation factor TIF1C. The termination of the rRNA synthesis occurs with the help of the factor called as TTF1, transcription termination factor, that binds and bends the termination site to the 3' end of the transcribed region. Thereby, it forces the RNA polymerase to come to a pause or the stop. So, the TTF1 is helping in the transcription release factor, that is further assisted by another factor called as transcription release factor, that is polymerase and transcript release factor called as PTRF. and a T-rich region that will induce RNA polymerase to terminate the transcription, thereby the RNAs are released in the form of a primary transcript. Here the diagram shows the binding up of TTF1 as well as PTRF for the termination of the transcription. This diagram shows the primary transcript of pre-mRNA that contains 18S, 5.8 and 28S rRNA. The primary transcript consisting of 3 different types of the spacer nucleotide. That is, one region of RNA is separated by another region by a special type of nucleotides, spacer nucleotide, that is not having any role. They are not going to be converted to any protein. So just dummy nucleotides, these are called as spacer nucleotides. There are 3 different types are there. What are all they means? NTS, ETS as well as ITS. NTS means non-transcribed spacer, ETS means external transcribed spacer, and ITS means internal transcribed spacer. These are further processed by exo as well as endonucleolytic cleavage guided by snRNA, that is smaller nuclear RNA, and these spacer nucleotides are eliminated to form the mature RNA, that is 18S rRNA, 28S rRNA as well as 5.8S rRNA. So we can see the diagram. The rate of nuclear transcription is very high, and the many polymerases operates at the same transcription unit. They are actively responsible for the production of the RNA. From the micrograph we can see, electron microscope picture, we can see that a Christmas tree like appearance, how the RNA polymerases are actively engaged. RNA polymerase I is actively engaged in the production of the mRNA. The gene coding for rRNA except 5S rRNA are located in the nucleolar part. The rRNA genes are highly repetitive in nature, and the eukaryotic cell contains how many copies? Means the primary transcript. 100 to 2000 copies of our rRNA is present per cell. Don't forget. How many copies are synthesized? Means 100 to 200 copies are synthesized. That is how it looks like a Christmas tree like structure. Once the primary transcript is synthesized, the snRNA is guiding. It cleaves them into individual 18S, 28S as well as 5.8S rRNA. Here the beauty is there is no cap and tail. Don't forget, it present only in the case of the mRNA. The rRNA genes are non-transcribed spacers, as I told. Here we can see that the same diagram. How these spacer nucleotides are eliminated. The last but not least is nothing but our RNA polymerase III. RNA polymerase III is responsible for the production of small non-coding RNAs including tRNA as well as 5S rRNA and U6 snRNA, SRP RNA, that is small ribonuclear protein RNA, that is snRNA, various snRNA, and other short stable RNA such as ribonuclease P RNA. All these are synthesized by the RNA polymerase. The different types of promoters are found in the case of the RNA polymerase III. Type 1 promoter. Here is an example of Xenopus levis. It's a toad The 5S RNA gene of the internal control region, that is ICR, is shown here. A, EIC and C region, and which is further subdivided into A box, and which is 50 plus 50 to plus 60, and an intermediate element IE, which ranges from plus 67 to plus 72, and a C box between plus 80 to plus 90. This is the location of this type 1 promoter complex of the Xenopus levis, and the type 2 promoter elements of Xenopus levis of tRNA contains the gene consisting of an A box plus 8 to plus 19 and a B box of plus 52 to plus 62. The type 3 promoter of the Homo sapiens, that is human being, of U6 sRNA gene consisting of distal sequence element, that is DSE, that ranges from minus 215 to 240, that enhances the transcription, and a core promoter composed of proximal sequence elements, that is PSE, that is minus 65 to minus 48, and a data box of minus 32 to 25 in the case of the tRNA promoter. The 5S rRNA and tRNA genes have a promoter within the coding region of the gene. It's an unusual. It is found in the case of the coding region. The promoter's region is from of 7S and U6 snRNA, and more or less like looks like the RNA polymerase II promoter with little differences. Though the size of the gene is small region ranging from 160 to 400 base pairs, their promoters are well defined, and transcription initiation from the respective start sites and as well as start sites in the promoter region. How the initiation of transcription in the 5S rRNA that takes place means the RNA polymerase comes and joins to the promoter region. It's an unusual. It requires no controlled sequences are found in the case of the upstream region. Instead, it normally relies on the internal control sequence transcribed within the sequence found in the transcribed section of the gene, that is C box, that is bound by TF3A first, which recognizes the C box. Then TF3B comes and binds, which contains data box binding promoter protein, and TF3A at the position of the start site. That is, A is recognizing, followed by B, and it melts. The TF3A melts the position of the start point. Then the RNA polymerase III complex associates and starts the initiation process in the 5S rRNA gene by the RNA polymerase III. Whereas in the initiation of the tRNA gene, the TF3C transcription factor polymerase 3C binds to the intragenic position, that is lying within the transcribed region and the control sequences, that is A box and B box. The TF3C, after binding it, it attracts the RNA polymerase III and starts the process of transcription from the start point. Once the primary transcript is synthesized, the initiation of the transcription of U5snRNA, m-RNA activating protein complex comes and joins to this position and enabled in the DNA opening and starts the transcription initiation of the U6 snRNA gene. The U6 snRNA gene is responsible for our splicing mechanism. The termination. Normally, the polymerase III terminates the synthesis of 5S rRNA, tRNA, as well as U6 snRNA gene by a small poly T stretch. So once the poly T stretch comes, automatically it comes to an termination process in the case of the eukaryotic system. All these pre-tRNA are processed by removal of the introns. Some tRNA precursors contains introns in the nearer to the anticodon regions. These introns are spliced out during the process of tRNA. All the tRNA, mature tRNA, contains CCA terminus at the 3' end. What is the difference between prokaryotic tRNA and eukaryotic tRNA means? Prokaryotic system tRNA devoid of CCA terminus, whereas eukaryotes contain CCA terminus at the 3' end. These bases are added by, not encoded by tRNA gene, but this is specially added by a nucleotide CCA transferase that is responsible for the addition of this. So this is added by the special mechanism. The mature tRNA, when the primary transcript of tRNA is synthesized, it doesn't have the special nucleotides like for example unusual adenosine, that is special adenosine, and dihydroxyuridine as well as pseudouridine. All these are not present. So normal nucleotides are converted to these unusual form, that is dihydro, that is TCU as well as pseudouridine. All these are thymines are unusual nucleotide which is found in the case of the RNA. This is added. How it is converted means by the unusual mechanism that adenosine, guanine, cytosine and uracil are modified at the tRNA to produce a special bases. This biological function of these modified bases is uncertain. Why this happens in the case of this? It's not clear. Here is the mechanism which shows. This diagram shows the primary transcript of RNA. The first step, the upstream sequences, the unwanted nucleotides which are present in the 5' end is eliminated by RNase P, that is creating a cut. That unwanted nucleotides in the region is eliminated. Then RNase D creates a cut at the 3' end to remove the nucleotide to create the 3' hydroxyl group. Once the 3' hydroxyl group is found by the RNase D, then comes the addition of CCA. The splicing of the introns are taking place which are found nearer to the anticodon arm. Then the fourth step, the CCA terminus is added to the 3' end of the eukaryotic tRNA. Normally this CCA terminus is absent in the prokaryotic tRNA. Once this is added, the basis modification is taking place and we can see the guanosine residue is methylated at DHU arm and thiamine is added in the T pseudouridine modification of the basis that takes place, methyl adenosine formation in TUC arm, and thiamine is added. Pseudouracil is also added to the TUC arm and we can see the anticodon arm. And this mature m-RNA, mature tRNA is consisting of all these things with the CCA terminus at the 3' end. Using this anticodon arm only, it may read the message which is found in the case of the m-RNA. It is converts the nucleotide language into amino acid language. We call it as polypeptide synthesis or protein synthesis, that is translation. That's all about the splicing mechanism of, that is removal of intron, how the split genes are modified, that is how they are converted, how the m-RNA, tRNA and rRNA is synthesized. This removal of introns and modification are called as post-transcriptional modification of RNA. So you may be asked separately for mRNA, rRNA or tRNA, or altogether post-transcriptional processing of RNA means you have to write all the three in your examination. That's all. Thank you.